Hemoglobin is like your body's mailman for oxygen. It has four subunits, and each of them can grab onto one oxygen. And it travels around the body and it picks up oxygen in your lungs and then it goes and delivers it throughout the body. Just like your mailman needs to not only grab the mail, but give it to the people, hemoglobin needs to grab the oxygen and crucially, it needs to then be able to let go of that oxygen. So it's got these two kind of contradictory tasks. And so we need to be able to actually change the affinity of hemoglobin for the oxygen so that it can both pick it up and let it go and do that in the right locations. And there's some really cool ways that biochemically this happens, including things like positive cooperativity, allosteric regulation, and effects such as the Bohr effect, which relies on pH, and all of this comes down to some really cool biochemical phenomena. So we're going to talk about all of these different things. And so first we need to, though, get some of the basic terminology out of the way. So let's go back and think about just binding in general. So remember that we could say, okay, we have A and B. They can bind, which is going to be your K on, and then they can come apart or dissociate, and that would be represented by your K off. And if you take that K off and you divide it by your K on, you get a value called the KD. The KD is equal in concentration to the amount of one binding partner you need to have in order for half of the other partner to be bound at equilibrium. So it's an inverse measure of affinity because the Ka would be the binding affinity. The Kd is the inverse of that. And so a smaller value is going to be a higher affinity, meaning that it's going to take less of the binding partner around in order for half of it to be bound. So we take one partner, we increase its concentration with a set amount of the other partner. As we increase the concentration, we add more encounters for there to be those binding opportunities. And so depending on the affinity, it's going to take more or less of those chance encounters, those bump-ins, in order for half of the other partner to be bound. So the stronger the KD, the higher the affinity, the bigger the KD, the lower the affinity. If we think back to our marriage analogy, and we think about instead of ligands, our binding partners are going to be people marrying. If they have a higher affinity for one another, they're more attracted to one another. They don't need to have as many copies of the other person around in order to find them married. And this could be because they are more likely to get married or because they're less likely to get divorced. And then the concentration determines how likely they are to meet. So the higher the affinity, the lower the KD. The lower the affinity, the higher the KD. Note that this is a linear scale, so we have a linear increase in the concentration of A. But we're often going to see things on a log scale or a semi-log scale, where we have one of the axes be in the log. And this is going to look like a sigmoid on a log plot. And it'll get confusing. But if you have a graph and you have a linear scale, often what's going to happen is when you're doing your experiment, you're going to want to have a large range covered. And practically the way that you often do this is by doing a serial dilution. So you take the concentration, you take your solution, a concentrated solution, and then you dilute it, say in half, and then that in half, and that in half, and that in half. And you end up with a lot of points that are scrunched really close together. On a log scale, they're farther apart from one another, they're nicely spread out. But now, if you have this hyperbolic curve, you're going to see a sigmoid. And I want you to keep in mind that you always need to look at the x-axis. Because if you have a sigmoid on a linear scale, that's going to tell you about something called cooperativity, potentially. And so a sigmoidal curve on a linear plot indicates cooperativity, which we'll talk about what is in a second. But if you see a sigmoid on a semi-log plot, that does not indicate cooperativity. So what is cooperativity? Basically, we have cooperativity, we have the potential for cooperativity if we have multiple binding sites. That doesn't ensure that we'll have cooperativity, but if you have a single binding site, you're not going to have cooperativity. So if you have a single binding site and you see a sigmoid, you should be like, oh, that is probably a log scale. But if you have multiple sites, then it gets confusing. And you need to make sure you're looking at a linear scale if you want to see based on the graph, easily see whether or not it's cooperative. And so cooperativity, this is where binding at one site affects the binding at another site. So you have to have multiple sites, and the binding of the thing at one of the sites influences the binding of the things at another site. Thinking back to our marriage analogy, it's like maybe you have a group of friends and they're trying to decide whether or not to get married. And you have one friend that gets married, and maybe they go and they say, "Oh, this is the best thing ever. You should get married too." And so then their friends are more likely to get married. That would be like what we call positive cooperativity, where binding of the first thing increases binding affinity of the next. Conversely, you could have something happen where the friend gets married and it's an awful experience for them. They're like, never get married, don't do it. It avoid it at all costs. And so then that next friend would be less likely to get married. That would be an example of negative cooperativity. You could also have a non-cooperative binding situation where the marriage of one friend does not affect at all the marriage of the other friend. So you have kind of these multiple possibilities. If you have multiple sites, you have the possibility it's cooperative versus not cooperative. And you can look to see on a linear plot, you can look to see if you've got that sigmoid shape, you've got cooperative. If you don't have cooperative binding. Negative cooperativity looks a bit confusing on a graph, so we're not going to focus on that. And we're going to focus instead on being able to identify the positive cooperative binding from one of these graphs. So we have the binding of one thing increases the binding of the next, and so you'll see the kind of inflection point in the sigmoid where you're going to go from a lower affinity state to a higher affinity state. And this is what we're going to see classically displayed in hemoglobin. If you want to get mathematical about it, there's actually this thing called a Hill coefficient, and there are these hill plots, and there's these mathematical equations, but they can be fooled, and so I'm not going to have you worry about hill plots. Just know that if you have this hill coefficient that's greater than 1, that indicates potential positive cooperativity, but not necessarily. And if it's less than 1, it indicates negative cooperativity, but not necessarily. And if it's 1, there's no cooperativity. Things get tricky though when they're like close to 1 and all this stuff. And so yeah, so don't worry about hill plots, but know that that's something that you might see is something called with that like an NH, and that would be telling you the hill coefficient. And you're welcome to go and learn more, and I will post a link. Let's go to hemoglobin. So hemoglobin, you've got these 4 chains. And so we have a heterotetramer. So remember, we can talk about oligomers or multimers where they have multiple chains, multiple protein chains. And we can classify them based on how many chains they have and whether those chains are the same as one another or different from one another. If we have a homomer, then they're all the same. And if we have a heteromer, that means that at least one of the chains is different than the other chains or another chain. And we can classify them based on their number. So here we have 2 alpha globin chains and 2 beta globin chains. So we have 4 chains total, so we call it a tetramer. And these chains are not all the same, so we can call it a heterotetramer. Each of these chains is going to hold on to oxygen, but it's going to hold on to them indirectly. Instead of binding directly to the oxygen, the hemoglobin is going to bind to this heme molecule, which is where we get the name hemoglobin. And so it binds to this heme molecule, and then the heme molecule contains an iron. And that iron is going to then bind to the oxygen, and then that's going to allow the hemoglobin to hold on to it. The protein, as we'll see, there's especially these like histidines are going to make interactions with the oxygen and with the iron. And that's going to help to make it so that this oxygen is going to be held in each of the 4 subunits of hemoglobin when you have the hemoglobin fully loaded. The heme group also absorbs some visible light, but it doesn't absorb red light, and so that's why the blood looks red, because it's absorbing the other stuff and letting the red light get like reflected back at you and stuff. That was what hemoglobin actually looked like, and we'll go and look in 3D in a minute. But for now on, for simplicity's sake, I'm going to draw it like this. So I colored it so you can just tell apart the different chains, so alpha and beta. But these are just copies of the same. So you have 2 of the same alpha chain and 2 of the same beta chain. And this is with adult hemoglobin, as we'll talk about with fetal hemoglobin. So Fetuses have their own form of hemoglobin that's better suited for picking up the oxygen from their mother's blood, which is the mother's already kind of like used the oxygen, so there's lower concentrations. And so they have a different version of hemoglobin. It has a gamma chain instead of a beta chain, but both of them have alpha. But for now, let's just focus on the adults. So sometimes we abbreviate this HbA, and this is going to have two beta chains and two alpha chains. It's the beta chain that we were talking about before when we talked about sickle cell anemia, because mutations in the beta chain can cause sickle cell anemia, as well as beta thalassemias. So we have those four sites, and we mentioned that it had positive cooperativity. So remember, if one oxygen binds, then what happens is that it increases the affinity of oxygen on these other sites. So if one oxygen binds, then it's easier for the rest of the sites to bind to oxygen. So although it might take a while, it might be hard for that first oxygen to bind, well, once it binds, then these other guys follow. But then the reverse is also true because your curve goes one way, it also goes the other way, right back. And so if an oxygen comes off, well, then the others are quickly going to drop off as well. So why is this useful? Well, if we take a binding curve for hemoglobin, binding to oxygen, here on our hemoglobin binding curves, you're going to get used to looking at something like this. On our x-axis, we have the concentration of oxygen in terms of its partial pressure. So this PO2. And then we have on our y-axis, we have the hemoglobin saturation. And this is referring to like all the copies of hemoglobin. So if we have no hemoglobin, none of the copies of hemoglobin have oxygen bound, we'd be down here. And then if we have all of the hemoglobin copies be bound to oxygen, we'd be all the way up here. And then at some point, you're going to have half of the binding sites bound. So you're going to have 50% saturation. This doesn't mean that you have like all of the hemoglobin copies have two oxygen bound. Instead, because of that positive cooperativity, that makes it so that when one oxygen molecule binds, the rest bind. And when one unbinds, the rest unbind. You're going to end up with probably like a 50-50 mixture of all bound or no bound. But half of the hemoglobin sites that are available to bind to oxygen are bound to oxygen. So you got 50% saturation. The partial pressure of the oxygen, so the concentration of the oxygen that got you to that 50% saturation, is your P50. So think of your P50 like your KD. And same applies. The smaller the value, the better the affinity. The larger the value, the weaker the affinity. And you want to think about shifting the curve to the left or the right. So left would be higher affinity, right would be lower affinity, just like you would see with the KD curve. The only difference is kind of what we call things and the fact that we're in partial pressures, which is indicated by this like P. Here we're seeing this evidence of our cooperativity. This is not a log scale, this is a linear scale. So we see that the sigmoid actually will tell us that we do have cooperativity. And if we go this way, we quickly increase the binding of oxygen. But then if we drop off, we got to drop off right back. And so if we want to think of what's happening at a molecular level, cooperativity, when you bind to one thing, well, then the others more quickly bind, the affinity increases. But remember that things are going to bind and unbind and bind and unbind. And even if your affinity is now high, you're eventually going to drop it. And so if you drop it, then what happens is quickly things reverse themselves. And so that's why you can kind of have, even at a higher affinity, you're still able to sometimes drop it. And when you sometimes drop it, then oops, they all go off too. But it also means that we can quickly increase the hemoglobin saturation for patients in the hospital and things like this if we increase the oxygen concentration. That'll make the initial binding more likely so that we can make full binding more likely. So there's a bit of debate and some theories about whether the binding happens in a sequential manner or whether it happens in a concerted all or none model. And so you might see things like MWC or Koshland. You don't need to worry about these, just know that there are a couple different models and it's not really clear. What is clear, however, is that there are allosteric effects taking place. Allosteric effects are where something that's happening at one place of a molecule affects another part of the molecule. So the reason why you're able to have these one change in one subunit affect changes in another subunit, we'll get into the why, but we can call that sort of thing an allosteric effect. So it doesn't have to be between different chains. It could also be within a same chain. Something happening at one place causes some sort of change, some sort of maybe conformational change, so a shape shift that is going to affect somewhere else in the protein or the molecule if it's not a protein. So that's allostery. Another couple words now. Modulator. When we talk about a modulator, that is a ligand that causes allostery that affects the binding of other ligands. So remember, ligand, that's just a generic word for a binding partner. And what we're saying here is that we have a binding site that causes allostery, so it causes some sort of change in the protein that affects the binding of something else. And this could be negative, where it decreases the affinity of the other thing, or positive, where it increases the affinity. So when we talk about hemoglobin, we can say that oxygen is acting as a positive modulator, causing allostery that affects the binding of further oxygen molecules, and it's doing so in a way that increases the affinity. We can also classify oxygen as homotropic. So a homotropic modulator is going to affect binding of the same ligand. So if you have multiple sites for the same ligand and binding to one of the sites impacts the binding of that same ligand, like another copy of that same ligand at a different site, we call that homotropic. So that's what we have here is we have oxygen is acting as a positive homotropic modulator. The opposite would be a heterotropic negative modulator. So hetero, different. So a heterotropic modulator is where a modulator affects the binding of a different ligand. So your modulator is not the same as the ligand. Whereas with homotropic, it is the same as the ligand. I mean, it's like a different copy of the same chemical molecule. So now that we got some terms out of the way, let's go and look at the effects of this allostery from a physiological perspective. Like, why is this important? So let's go back to our mailman analogy. And so you've got your mail in your mailroom, and there's a bunch of mail in the mailroom. And then there are people all around who need mail. And so similarly, you've got a bunch of oxygen in your lungs, and then you've got a bunch of tissues all around your body that need oxygen. And the oxygen concentrations are low over there, and then they're going to be high in your lungs. As we'll get more into, the ability of hemoglobin to kind of have this cooperativity is because it's able to transition between two states, so two conformations. One of the conformations is this oxygen-loving R form, and the other conformation is this not so oxygen-loving T form. As we'll talk about, the T stands for taut because there are salt bridges that are actually holding this form of hemoglobin in that conformation that doesn't want to bind oxygen. So T for taut and R for relaxed. And I like to think, well, relaxed, you've got oxygen, it's relaxed, you're relaxed, right? So the relaxed hemoglobin, the R form, has the oxygen. It's got a high affinity for the oxygen, so it's holding on to that oxygen tightly. But then when you have a low concentration of oxygen, and for other reasons, we switch to this T form, and the T form has a lower affinity for the oxygen. So we've got these two forms of hemoglobin, and they're really just different conformations of that hemoglobin. So same molecule, just underwent a shapeshift, one of those conformational changes. So that is what we're seeing with this curve that looks like a sigmoid. So see, we've got a linear scale, and we see a sigmoid, so this is telling us this is a cooperative curve. Whereas these curves, so this one right here, and this one right here, these are non-cooperative. These are not showing cooperativity. These would be what would happen if you were always in a low affinity state. So if you're always in that T state. Whereas this curve represents what would happen if you were always in the high affinity R state. And what's the reality is that we're in this middle curve where we have this cooperativity, where we have this transition from a low to a high affinity state, depending on the oxygen concentration. So here, if we're looking at a graph like this, we have our oxygen pressure of oxygen in our tissues and then in our lungs. And so you can see in our lungs, we have a lot of oxygen and our tissues, not so much. What you want to do is compare the proportion that's bound in the lungs to the proportion that's bound in the tissues, and this is going to allow you to see how much is picked up on the if you go from left to right or dropped off if you go from right to left. So if we take the oxygen concentration in our lungs, we're up here close to about a little over 12, and if we're down in our tissues, we're closer to 4. If we compare what would happen if we had no cooperativity or we had cooperativity. So I'm not even going to show lines for this high affinity because you can see it doesn't change, so we wouldn't be dropping off anything. What if we were stuck in the low affinity state? Well, here in our lungs, we have about half of it bound, and then we have about 0.2-ish in our tissues. So we've released about 20% if we were stuck in this low affinity state. But what if we were in this cooperativity? Well, now we get to release about 38% because we're going closer from about zero point 0.6 to about 1. And so, see, in our lungs, we've got it almost fully bound. Whereas if we were in that low affinity state, we wouldn't. We would only have about half of it bound. But if we were in the high affinity state all the time, well, then we never let it go. So by having this cooperativity, we're therefore able to release an optimal amount. Let's look a little closer at the actual basis of this cooperative movement. So inside each of those subunits of hemoglobin, there is a bound heme, and that heme has a bound iron. And that bound iron can bind to oxygen and let go of the oxygen. How this heme is held in place is in part through these histidine residues on either side of that heme group. One of these histidine residues is going to make contact with this oxygen, so bind to this oxygen, and then the other histidine residue is going to bind to the iron. And so this way you kind of hold it from both sides. When oxygen is bound, it changes the conformation. It pulls on the heme, and so it causes it to go from this puckered state that you see for the heme and the T state to this kind of linear state that you see in this R state. So you get this kind of yanking of this heme group when you bind to that oxygen. Not only are you yanking the heme group, but remember that the histidine was making contact with that heme group through the iron. And so now you're yanking the whole protein. And not only does that affect this subunit, it's also going to affect the other subunits. Because when you have a change happen at one of the subunits and it's connected to the other subunits, those other subunits are going to change as well. And not just locally, but they're also going to change kind of in their interfaces. And this is going to cause it so that when you go from this T state to the R state, you're bringing out of contact the amino acid residues that actually form salt bridges that connect the alpha and the beta subunits. And so you can see that you have this lyse and this his, and so this asp, so you have these basic and acidic amino acids that are normally going to be forming ionic bonds in this T state, but when you bind to the oxygen, you yank those apart and you can no longer form those interactions. So you can see that these amino acids are kind of pulled out of position. And so what happens is that when the oxygen binds, you get this conformational change. The oxygen yanks on the chain, that chain yanks on the other chains, and as a result, you break off those ionic bonds, those salt bridges that were holding things in the top form. So it's a little confusing because in the top form, it actually looks a little like less taut in some ways because it has this kind of bigger central opening, but it's taut because it has these salt bridges in between the different subunits that are stronger. And keep in mind this bigger opening in the top phase because this is actually going to allow for a regulatory molecule called 2,3-BPG to bind. And that 2,3-BPG is going to actually be what's responsible for a large amount of the ability to have this cooperativity. I really like this animation. You can go through and click on the different views. And so you can see that you have this histidine on one side that's bound to the iron through the oxygen, and histidine on the other side that's just bound to the iron. And the iron is being held inside of this porphyrin ring of the heme. When oxygen binds, it kind of yanks the ring, so you go from a puckered to a flat. If you click on the different links, you'll see, so now we have in space filling, and now we're going to see it in the context of the tetramer. So we've got those 4 chains. So you can see we've got oxygen bound. So we were R, and now we're T, R, and T. You have those movements of the heme group that are then going to cause the subunits to change. So conformational changes, and those conformational changes are going to affect the interactions that can form between the different subunits. And so in particular, you can see that this move from the oxy to the deoxy, we're moving the beta chain C terminus. So this histidine C terminus, so the C terminus is negatively charged. We've got this lysine here. This lysine can then form a salt bridge with this histidine. And this aspartate is going to be important for holding this histidine in the right conformation. And when we talk about the Bohr effect, we'll see that this histidine needs to be positive in order for this aspartate to form this salt bridge that orients the C terminus to form this interaction with the lysine. And so that's why at a lower pH, you're going to have more proton availability. The histidine is more likely to be protonated. So you're then going to get the aspartate interaction that can then allow that C terminus to bond to the lysine. So we'll get more into that when we talk about the Bohr effect. As we talk about, there's a lot of different ways you can represent structures. And so here we're seeing kind of space filling, and then these are shown in stick. And it's indicating the distances between these amino acid atoms that are interacting. Note that we're going to be moving when we go from the R to the T, we're kind of opening up a spot where the molecule of 2,3-BPG is going to bind. And 2,3-BPG is only able to bind there when it's open and it's closed in the R form, but it's going to be open in the T form. And when it binds in the T form, it's going to be stabilizing the T form, and therefore it's going to be stabilizing that form that has a low affinity for oxygen, and it's going to make it hard for oxygen to get picked back up. So there's a lot more in this tutorial, and I encourage you to go check it out and play for yourself. Thanks to Eric Martz and Frieda Reichman. And so this is just a free tutorial out there, interactive, very, very helpful. So let's talk about this 2,3-BPG. So first of all, what is it? So 2,3-BPG, 2,3-bisphosphoglycerate. Sometimes you might see like disphosphoglycerate, so it might be abbreviated DPG. And what it is it has 2 bis. So when we see bis, that means 2, as opposed to di. So if it was di, it would be like 2 phosphates next to one another, like ADP, adenosine diphosphate. It's got 2 phosphates like attached to one another. When you have bis, that means they're in separate places. And so this is 2,3-bisphosphoglycerate. In terms of where it comes from, so 2,3-BPG is actually made from 1,3-BPG, which is when we talk about glycolysis, that's going to be one of the key intermediates of glycolysis. So with 1,3-BPG, they're on opposite sides of this 3-carbon molecule. With 2,3-BPG, they're next to one another. In order to shift them next to one another, you need to take a detour from glycolysis. So there's this enzyme bisphosphoglycerate mutase that will then go and shift things around, and then you can change things back to get 3-phosphoglycerate like you would get from glycolysis, thanks to this 2,3-bisphosphoglycerate phosphatase. You don't need to know this pathway. This pathway does have a name though, the Rapoport Leubering Cycle. And it's energetically expensive because you bypass a step in glycolysis that would actually make ATP. And yet, about 15 to 25% of the glucose that gets converted to lactate in your erythrocytes, so your red blood cells, actually goes via this 2,3-BPG synthesis. So you make a lot of this, and you make a lot of it when you're doing a lot of glycolysis. And so, yes, you lose out on getting a little bit of energy, but it's so important because we need it for that hemoglobin cooperativity. And so a lot of this is getting made in your blood cells when your blood cells are being really active. So it's got these 3 negatively charged groups. And so this is going to be a very negatively charged molecule. And so we keep that in mind when we think about what we would want to hold it in place. As I mentioned, we have an adult and a fetal hemoglobin, and we're going to focus on adult hemoglobin right now, and then we'll get into fetal hemoglobin. If we look in the absence of BPG, We basically don't really see any cooperativity. If we do have BPG around, now we get that curve that we were looking at before. So when we were looking at those cooperativity curves, they were basically assuming the presence of BPG. Without BPG, now we're stuck in a high affinity state. So we're stuck in this high affinity state, which is telling us that the BPG is going to be acting as a negative allosteric modulator. And it would be a heterotropic modulator because it's affecting the binding of oxygen, but it's not oxygen. So negative because what's happening is it's decreasing the affinity. Allosteric because it's affecting somewhere else in the protein, it's affecting another binding. And heterotropic because it's not oxygen, but it's affecting the binding of oxygen. And it's decreasing that binding. Without me telling you that it's a negative modulator, you can see that the curve shifts to the right. The P50 for oxygen is going to be increasing when you have a lower affinity. So if your curve shifts to the right, that's a decrease in affinity. Whether you have cooperativity or not, if you shift the curve to the right, that means you need more of it in order to get the same amount of it bound. Or so you need more of it in order to get half of it bound. So when you see a shift to the right, think lower affinity for oxygen. Think negative modulator. And so BPG is acting as a negative modulator. It's shifting that curve to the right. So why is this? So going back to that T versus the R state, remember I said that T state kind of had that opening, that central opening? That is where the BPG binds. So it's not binding in the same place as the oxygen. So remember the oxygen is binding onto these hemes. That's not where the BPG is binding. So it's not directly competing with that oxygen, but its binding is decreasing the ability to bind to oxygen at those other sites. And this is because it's binding in that place that's only open in the T state. And so when the T state is open, it can bind, and then it kind of locks it into that T state. It stabilizes that T state. So remember that 2,3-BPG had those negative charges. And when we have negative charges, the best interaction we could have with them would be a positive charge. That would give us an ionic bond, one of those salt bridges. And in a protein, where can you find positive charge? Well, that would be our basic amino acids. So we've got our histidine, our lysine, and our arginine. If we go and we actually go and look at the binding site of the 2,3-BPG, what do we see? Oh, hey, histidine, lysine. So we've got these positively charged amino acids that are forming these salt bridges with the 2,3-BPG. So now let's go back and look at another one of those binding curves. And so this one looks a little more complicated. Apologies, the book kind of overlaid a lot of stuff, which can be a little overwhelming, so we'll take it one by one. Here we're seeing the oxygen in the lungs at sea level, and here we're seeing the oxygen in the tissues, just as before. If we focus on this blue line right now, so the black line is no BPG. The blue line is if we have 5 millimolar BPG, which is fairly typical, and then the green is 8 millimolar BPG, which as we'll see is going to be what you'd want at a higher altitude where there's less oxygen. If we look here, we see that by comparing this point to this point, we get about 38% delivered. If we were at this like sea level amount of oxygen, we pick up almost all of it in our lungs. We deliver about 38% of it to our tissues. So lots of oxygen gets delivered. If we started, however, at the high altitudes where we have lower oxygen concentrations, well, now you can see we're starting lower. Starting at different levels, but we're ending at the same level. And if we're starting at a lower level, we're going to deliver less. And so we'd only be delivering about 30% of the oxygen if we had that 5 millimolar BPG. Now let's think about what would happen if we increase the BPG concentration. So remember that increasing the BPG concentration, what it's going to do is it's going to shift our curve to the right, meaning it's lowering the affinity. If we lower the affinity, we shift our curve to the right. Now this steeper part of the curve is going to come into the range of the tissues. And so we have a little bit lower affinity when we have the top, at the top, but then we're going to shift it more down here. And so overall, we have just a tiny bit lost here, but then we have a bigger amount gained down here. And so we would end up delivering about 37% of the oxygen of the hemoglobin's oxygen capacity. And that's pretty similar to what we would have if we had started at sea level with the 5 millimolar. And so we're able to compensate for the decrease in the amount of oxygen by decreasing the affinity for the oxygen. So hopefully, I know it's a little weird, but you want to lower that affinity so you can offload more at those lower oxygen concentrations. So small decrease here, but big decrease here. The people who live in high altitudes will make greater concentrations of this 2,3-BPG, and athletes will sometimes go to higher elevations to train to get their bodies to start making more of the 2,3-BPG. So they have an advantage in terms of oxygen carrying when they're back at the sea level. So now let's get back to what I gave you a spoiler about earlier. So fetal hemoglobin. So fetal hemoglobin, unlike adult hemoglobin, which we abbreviate HbA, we abbreviate fetal hemoglobin HbF, and it has gamma chains instead of beta chains. So this is the Greek symbol for lowercase symbol for gamma. I know it's weird, it looks a little like a Y, but that's a gamma sign. And instead of the alpha chain, so instead of these blue chains, the fetus would have the gamma chains, which are very similar, but they have important differences. And these gamma chains stop getting made soon after birth and get replaced by the beta chains. And so after birth, you stop making the fetal and you start making the adult hemoglobin. You still have the instructions though for making that fetal hemoglobin. So for sickle cell anemia and beta thalassemia, where patients have problems with this beta globin, there are therapies and treatments and gene therapies and stuff that are used in order to increase the making of the fetal hemoglobin that got shut off, try to like switch it back on so that you can make that to compensate. But the fetal hemoglobin has some different properties. And so one is that it has a very low affinity for 2,3-BPG. So this is the adult hemoglobin without BPG that we saw before, and the adult hemoglobin with BPG. So this huge shift, this huge decrease in affinity with BPG, and this great cooperativity. Now let's think about the fetal hemoglobin though. So fetal hemoglobin without BPG versus fetal hemoglobin with BPG. So you can see that without BPG, it has a slightly lower affinity than the adult. But with BPG, it's got a lot higher affinity than the adult because the BPG has less of an effect on it. So remember, shift to the right, decreasing the affinity, shift to the left, increasing the affinity. And if we compare the adult hemoglobin with BPG to the fetal hemoglobin with BPG, we see that the fetal hemoglobin has a higher affinity. And we can see that this higher affinity is coming because it's less affected by the BPG. So it's less shifted by the BPG, which makes it have a higher affinity in the presence of BPG. So why is this? Well, it has a higher affinity for oxygen because it's got a lower affinity for the BPG. So BPG, it was binding to that central cavity of the adult hemoglobin through those salt bridges. Those salt bridges were involving amino acid residues from the beta chains. Those beta chains are not present in the fetal hemoglobin. Instead, the fetal hemoglobin does not have all those basic amino acids that are needed to make those interactions. So you can see that you've substituted histidines for serines, and serines are not going to be able to make those salt bridges. So you're not going to be able to form those ionic bonds with the BPG in the fetal hemoglobin. As a result, the fetal hemoglobin has a lower affinity for 2,3-BPG and it gets shifted to the right less. Why is this important? Well, let's do again our comparison of different concentrations and different oxygen concentrations and the amount of the oxygen that would be bound, and therefore the amount that would be released. So now, though, instead of looking kind of in sea level versus our tissues, or at sea level versus high altitude versus our tissues, we're going to be looking at adult venous blood versus fetal arterial blood and fetal venous blood. So adult venous blood, this is what the baby's going to be seeing, or the fetus is going to be seeing. And that's going to be after it's already basically been used up by the mom. So the mom goes and they take their share. And now there's still this little bit left over. And so now we've got to say, okay, can the baby take that? And so in order to take it, you need to have a high affinity. And if you had a lower affinity, you wouldn't be able to take it. So this purple line is going to be the adult, and this green line is going to be the fetal. And this MB, that's myoglobin, and so it does not exhibit cooperativity because it's monomeric. So there's only one subunit, so you can't have a finding of one thing affecting the binding of another subunit because there's only one subunit, only one binding site. And so what you're going to see here is going to be that the adult hemoglobin, it would have basically not be picking stuff up because it's going to be dropping it off. And of course, it's in a dynamic equilibrium, so you're always dropping and picking up and dropping and picking up and dropping and picking up. But in this case, you're much in favor of the dropping and leaving. And if you pick it back up, you're going to drop it right back. And so the adult hemoglobin has a really low affinity, and that would not be useful for the fetus to be able to pick up enough of the oxygen. But now if we look at the fetal hemoglobin, well, now we can see that at the concentrations in the adult blood, we're almost to 50% of the hemoglobin of the fetal hemoglobin being bound to oxygen, about 50% of the sites. And so we've seen that we're shifting this curve to the left compared to the HbA. And so we have a higher affinity, we're binding to a greater proportion of the oxygen that we're seeing. So in the adult, we wouldn't be binding to very much of it, but in the fetus, we are. And now we say, okay, we're starting with more. That's great. But before we said if we started with more, then that was going to cause a problem with the altitude. But the problem with the altitude was that there was still a lot of oxygen, but you couldn't deliver it well. Here, there is going to be not very much oxygen, and so we need to increase the affinity for that. And then when you get at these really low concentrations, even if you still have a fairly high affinity, what's going to happen is that if you drop that oxygen, there's not enough oxygen around in order to find one to pick back up. So you're still going to be able to release enough oxygen. And so if we go and look at the fetal venous blood, which is where the oxygen would kind of be released, you can see that in the case of our HbA, we would not be releasing very much. But in the case of our fetal hemoglobin, now we're releasing a large amount of the oxygen. So, to review, allostery is where we have something happening at one location of a molecule, such as a protein, affecting something happening elsewhere in the molecule. When we have a molecule that has multiple ligand binding sites, we can have the possibility for there to be cooperative binding, where the binding at one site influences binding at other sites. This can be positive cooperativity or negative cooperativity. And we can see evidence of it if we see a sigmoid on a linear plot, but not if we see a sigmoid on a log, semi-log scale, because even a non-cooperative binding would show as a sigmoid on a semi-log scale. The molecules that can affect binding at other sites are called modulators, and modulators can be homotropic, so they affect the binding of a same ligand, or heterotropic, where they affect the binding of different ligands. So we can classify oxygen as a homotropic modulator of hemoglobin that is a positive modulator. And we can classify 2,3-BPG as a heterotropic modulator that is a negative modulator. We can see effects of modulators if we look to binding curves, and the P50 is similar to the KD, but it's in terms of gases. And so we can see evidence of cooperativity in hemoglobin saturation curves, and we can see changes in that affinity of the hemoglobin when modulators are around by looking at where the curve shifts to the right or to the left. If we have a negative modulator, it'll shift things to the right, which is the case with 2,3-BPG. But if we have a modulator that increases the affinity, we'd be shifting to the left. 2,3-BPG, as we said, is a negative modulator, and it seems a little counterintuitive, but you want to increase the concentration of 2,3-BPG at high altitudes where there's less oxygen available in order for you to deliver more oxygen. Conversely, fetal hemoglobin has a lower affinity of 2,3-BPG, and so it has its affinity decrease less than the adult hemoglobin. So overall, you kind of have a higher affinity in the presence of BPG in the fetal hemoglobin. That higher affinity allows it to pick up oxygen when there's not that much in the blood that it's exposed to, but then also drop it off.